This is the first of a two-part lecture on recombination. And I'll start by saying that there are two fundamental types of recombination. Site-specific, which involves the use of a very particular sequence in DNA that's recognized, and recombination will occur at that site. And homologous recombination that requires only that two DNA sequences have reasonable homology with one another, and you can get a recombination event there. There are a number of different important enzymes that can mediate site-specific recombination. So today at this point, we're only going to be talking about site-specific. We're going to deal with homologous recombination in the upcoming talk. We're going to look at the Hin-Recombinase, and it actually is identified as an invertase, and you'll see why it's called an invertase, because DNA will be inverted. We'll look at some Tyrosine recombinases, enzymes that carry out recombination, again, why we have this ASE ending, that means it's an enzyme. And obviously these have an active site tyrosine that is involved in this process. We've seen some of these earlier, tyrosine, enzymes with tyrosine at their active site when we were talking about transposition, and earlier we talked about XRECD, which is involved in separating catenated chromosomes. We'll look today at CRE. There's an integrase. We will not talk about integrases, but you need to know that integrases are often used by viruses, again, to integrate viral DNA at a specific site into a chromosome. And then finally, resolvases. We've also looked at resolvases, and res is the example that should be familiar to you. Well, let's look at some of the specific examples. These are two categories of recombinases that have an active site, serine. And notice that they're very different in size. The first group includes a resolvase, res, that we've already seen that functioned in TN3, and it's very similar to the Hin invertase. So we're going to be talking about Hin in just a minute. This is the active site, active region of the enzyme, and this blue box indicates that this is the region that's conserved in many of these types of enzymes. And notice that very near the N-terminus we see this active site serine. Here's a larger version of a serine recombinase, and we see that in a number of transposons that we talked about a little while ago, and also it's involved in a recombination event that's responsible for sporulation in the organism Bacillus subtilis. And that is talked about later in the textbook. I'm going to look first at one of the examples in Anabaena, because the recombinase there plays an important role in heterocyst formation. Well, let's look at an example of how these types of recombinations, site-specific recombinations, really change the fate of some of these organisms in the environment. So in this picture we're looking at an organism called Anabaena. And this is a very important photosynthetic organism of the cyanobacteria group. It's very prevalent out in the ocean. You'll recall that cyanobacteria really are responsible for the evolution of oxygen on the planet, and these organisms are still major players out in the ocean, in particular to produce oxygen for us. And of course they're a beautiful green color because they're photosynthetic. So they need to carry out photosynthesis because that's how they get energy and that's how they fix carbon. But they also need nitrogen. So carbon and nitrogen are, two big things that organisms need. So if there's plenty of nitrogen, and the best form of nitrogen is ammonia, these organisms exist in these long bead-like chains. They're green, they're carrying out photosynthesis, and they're happy campers. but sometimes they run out of nitrogen. And when they do, they must start to fix nitrogen. Nitrogen fixation is the only way that they can survive if they're in a nitrogen-depleted environment. But the problem is that the only way that organisms evolved nitrogen fixation, meaning dinitrogen, in our atmosphere, was to convert it to ammonia, our favorite source of nitrogen, using an enzyme called nitrogenase. Now, here's the conundrum. Nitrogenase is very exquisitely sensitive to oxygen, and yet these cells need to produce oxygen because they're carrying out photosynthesis. So what this organism evolved is an amazing strategy to produce a differentiated cell. And it actually happens in a very particular pattern. So Anabaena makes a heterocyst approximately every 10 cells, so they can actually count. What this allows them to do is the cells in between will carry out photosynthesis and make carbon and energy, and the heterocysts will carry out nitrogen fixation and share the ammonia with the other cells. So it's a perfect kind of community. But part of this evolution of the heterocyst involved a DNA rearrangement, because all of a sudden now this one cell, spaced about every 10 cells, has to undergo a DNA rearrangement that allows it to start making this enzyme nitrogenase to produce new components that allow the cell to become very large and protected from oxygen, and also to shut off all the photosynthetic operations going on. And that's all carried out by one beautiful little DNA rearrangement. These recombinases all have slightly different mechanisms that they use to carry out the recombination, and I want to just look at one example for one of the serine recombinases. This happens to be the gamma delta recombinase, but it's very similar to the recombinase that works in some of the transposons such as TN3. So first of all, I want you to notice that there are 4 enzymes. Here is our donor DNA, and here is the target DNA. We've got 4 enzymes. Each one is binding to the recombinase binding element. That's what the RBE stands for. You'll see slightly different nomenclature in other papers. It depends a little bit on the field, but basically the enzymes are going to recognize a particular sequence in the DNA. And this active site serine with this OH group on the serine will be able to attack the phosphodiester bonds a little bit away from the RBE, and what that ends up giving us are these staggered ends. So we have now 2 sets of staggered ends, which, as you'll see, this is going to be important because now we're going to see a rotation between these enzymes as they flip over. And now that we've switched these strands, we're up here, and of course we've preserved these phosphoserial bonds. We still have 4 of those. Flipping the DNA didn't affect that. And now these OH groups, the 3' OHs on the DNA, can now attack that bond. Fortunately, the flipping occurs before that rejoining occurs, and we end up connecting those pieces of DNA together, but we now have this piece of DNA is joined with, the gray is joined with the purple, and the DNA that was on the top has now been joined to the bottom of the other strand. So we've just flipped the pieces of DNA. In the next few minutes, I'd like to look at a couple of examples of DNA invertases, and we're going to specifically look at how site-specific recombination plays an important role in virulence for a lot of microorganisms. We'll talk about bacteria here, but there are similar situations that occur in eukaryotic microbes. Now, before we talk about that, I want to just talk in general about phase variation, because this inversion is just one example of a larger process called phase variation. And phase variation really results in altered expression of different surface antigens in many organisms. In Helicobacter, the example we'll look at in a minute involves production of a different form of lipopolysaccharide, or LPS. Neisseria can undergo phase variation to produce a lot of different kinds of surface proteins on the cell, which will help them escape the host immune response. Some strains of E. coli produce different types of pili called pyelonephritis-associated pili. This is a modification or a phase variation that involves changes in the pattern of DNA methylation. We'll also go through an example where the H antigen or flagellin in Salmonella and organisms like Pseudomonas can be changed, and finally EPS and biofilm are also subject to phase variation in different organisms. And when I talk about this, I'm really talking about a situation where we've got a form of an organism, so let's say it's Salmonella, and we've got form A, meaning it's expressing certain components on the surface, and at a particular frequency, this organism will be able to switch to a different form. Now I want to say that many of the cells will still be in form A, but a subset of the cells will undergo a phase variation, and now we'll have form B. This is a reversible process, I want to point out. So form B can go back to form A at some point in time. Why this is important, of course, is because if the immune system initially sees form A and it mounts an immune defense, it takes a while for that to happen. By the time the immune system responds, it is going to be able to recognize form A, but if cells are also existing in form B by that time, they will be invisible to the immune system. So this is a really important process for pathogens. Now let's look at one example. How do you actually recognize this kind of phase variation? Well, here's a situation where we're looking at the lipopolysaccharide variation in Helicobacter, and they've taken colonies on a plate and they've stained them with an antibody that recognizes one form of that LPS. So they stain dark. See a couple of dark colonies. But notice there are a lot of colonies that are quite light. They do not have the form of LPS that's recognized by this antibody. And then you can see an example of phase variation in action because some of the cell colonies will sector. They started out as this light-colored colony that doesn't react with the antibody, but while this colony is growing, some of the cells have undergone phase variation, and now they are producing the form of LPS that is recognized by this antibody. Let's look at the mechanism of Hin-mediated inversion of the Flagellar promoter in Salmonella and see where that takes us. So most of the time, if you isolate flagella, you see that the cells are making a type of protein that makes up flagella called FLGB. So they're all red. And here's the FLGB gene, and this is being transcribed and translated because here is our promoter and it's facing in this direction. So we're making FLGB to make flagellin, and we're also making transcribing a second gene called FLGA, and FLGA is a repressor, and it goes elsewhere on the chromosome and binds upstream of FLI-C and represses transcription of FLI-C. Now, periodically, when the Hin protein is made, and notice that the gene for Hin is between these two purple boxes that I've drawn on the DNA. So Hin gets made, and what it does is it mediates a flip, and I'll show you this on the next slide, so that now instead of making Flig B, we're making Fly C. Now let's look at some detail of this. And before I go talk about that, I want to point out that this was work done by Kelly Hughes, who was at the University of Washington for a long time and now is at University of Utah. And he did this work when he was a postdoctoral fellow in Mel Simon's lab at Caltech. This type of inversion happens at a pretty high frequency, so about 1 in 100 to 1 in 1,000 cells will be undergoing this type of inversion to switch out the different flagellin at any given time. And let's look now at how that actually is going to transpire in the cell. So again, here's Flig B, and that was the situation that I just showed you. So our promoter is oriented in this direction, so we can transcribe and translate the Flig operon. So we get our subunit for flagellin, and we get the Fly A repressor. Now when Hin is made, it's going to bind to those purple boxes. I don't have a perfect match for the color. And what that does is it's an invertase. We've already established that. And it's going to invert this very specific region on the chromosome because it's site specific. It's only going to recognize those sites. Of course, knowing about those sites and knowing about Hin, you could move these any place on the chromosome. So this is a great tool, and the people that studied this in the beginning realized that they had something that they could use as a great molecular tool. But let's look at what's going on here now. P is facing the opposite direction, so it's still active. It can transcribe the genes over here, but notice that we're not going to make any mRNA for Flig B and Flig A, so that means we're not going to make this flagellin up here, and we are not going to be able to repress Fly C anymore. So that repressor is gone, and now the ability to make Fly C allows the cell to start making this new flagella on the cell. Let's look a little bit more at the Hin mechanism. This is a figure from your book. Again, Flig B gives us one flagellin. It's actually called H2. And these are designations that actually, when you see pathogens referred to, sometimes they'll be given an H number, and that refers to the type of flagellin that they make because it's very important. Fly C is actually called the H1 flagellin. But here's the business end of this, and we've got our two regions that are the binding sites for Hin. And again, notice that these are inverted repeats. We've seen this a lot before generically, and I gave you a question about this on a quiz recently. Here is the Hin gene in between those invertible regions, and the Hin is the promoter for Hin is active all the time, so we're always making some Hin protein. And Hin, as I said, will be able to bind to this and flip this whole region around, and that's shown here just to remind you how these inverted repeats work. The Hicks L and R, they're shown right here, and Hin can bring those together, and then of course just change this whole thing. So now we go from here over to here, back around and down, and that's how we flip this entire region, only between the regions that are flanked by that inverted repeat, and the inverted repeats just stay as they are. But now of course we have the Flig operon in the off position and the H1 Fly C in the on position. Now these things are conserved in a lot of different organisms. A number of phage contain regions that are very similar. In fact, they're given similar names. PICs and GICs, for example, in different kinds of phage. And there's a very similar mechanism that's seen in an organism called Photorhabdus that has a symbiotic relationship with a nematode. But when the nematode gets inside of an insect, so this is a really remarkable three-way kind of, I won't say it's a symbiosis, it's a symbiosis between the bacterium and the nematode, but when the nematode is hungry, it will infect an insect, and once inside the insect, the bacterium, Photorhabdus, undergoes a switching similar to what we see in Hin, and it activates about 10% different genes in its chromosome so that it can help the nematode kill the insect. It's really a remarkable process that involves this kind of recombination. And the bacterium is responding to the fact that it's in a nematode inside an insect. Well, let's look at an example of a tyrosine recombinase, and this example is going to be different than the serine recombinase that we looked at a little bit earlier. The example here is Cre, and Cre is actually a recombinase from a phage that typically binds to LOX-P sites, LOX-P. And even though we haven't talked very much about phage, this is a really well-known example and representative of a larger category of these kinds of enzymes. So in the very beginning, we're going to start by looking at these 4 subunits. So we've got 4 Cre subunits, 2 in purple and 2 in white. And notice that we're looking at cutting by only 2 subunits initially. So I've highlighted with the 2 red spots where the initial cleavage is going to be. 1 on each strand of DNA, and they're on opposite subunits, as you can see. Now we're going to start at the bottom where that cutting was here, and we're going to take the DNA that was there and move it over to the upper strand. So it basically flips to the opposite subunit, and the DNA that was, I'm going to switch colors for a second, the DNA that was up here on the top white subunit is going to flip down to the bottom. So they're really just trading places. They're going to be rejoined, and now we've gone from over here where we have 2 strands of DNA, we've moved to this cruciform structure, and the cruciform structure has been sealed so that we have a covalent backbone again on all the DNAs. Well, now we're going to undergo an isomerization. So what we're really looking at here is, notice that the thin part of the axis between the strands of DNA and this cruciform is kind of on the north-south plane. And now we're going to, when we do this isomerization, we're going to elongate that DNA so that it moves out like this. And now we've changed the position and the orientation of where the DNA strands are relative to one another. We haven't switched the subunits though, so that's an important distinction between this type of recombinase and the serine one that we looked at earlier. And now we're ready to cut the DNA again. Now initially we cut DNA in the 2 white subunits, and now the DNA is going to be nicked, one strand each, again, but it's going to be on the purple subunits. So we've got cleavage occurring on those 2 strands, and we're going to rearrange the DNA. So the DNA that was nicked at the top on the purple strand is going to move down here. So it flips down to the other strand where the DNA was cut. So the DNA that gets nicked here can shift and flip up to the other top Cree that is on the upper left-hand corner. And notice now we've gone from DNA that is forming a V shape top to bottom to the V's being side to side. And of course we rejoin all the strands and the enzymes will leave and we've undergone a recombination. Well, scientists are always looking for ways to take information that they have learned from basic research and use it to make genetic engineering easier. And of course this was an amazing system using Cree and LOX, very well defined. The mechanism had been worked out, and this LOX site is, pretty easy. You can add that and incorporate Cree into a plasmid or a transposon system, for example, and companies like the Jackson Laboratory, which specializes in making very specific mice that are used as model systems for people studying disease processes, have been able to use these to make inversions, translocations, deletions. They've made reporter strains, conditional knockouts. So this is another one of those tools that scientists have learned to use to great advantage.